Hello there, this is Rohan Mishram again, and in our previous lecture we dealt with the topic of how do we identify drug targets as such. Okay, so in our drug discovery protocol, in previous few lecture we have dealt with this topic in more detail, the topic that in which we covered how do we identify drug target. We have covered experimental techniques as well as computational or in silico methods that are implemented in identifying the drug target. For example, we have dealt with one of the computational method, like how you can use subtractive genomic method or reductionist approach for identifying the drug target. Then we have moved on to our next topic called pharmacophore modeling. In pharmacophore modeling, we have tried to study the concept of pharmacophore and what exactly is pharmacophore modeling. We have studied its application, like how this pharmacophore modeling can be used in drug screening, and we have also learned how pharmacophore modeling can be used in terms of pharmacophore alignment. We have considered an example of CDK2 inhibitors, and finally we dealt with how this pharmacophore modeling can be used in identification of drug target. For this we have used I have already mentioned you that drug target can be identified using this pharmacophore mapping approach, which is implemented in software called as PharmMapper. It is an online free tool which take input the structure of your ligand, 3D structure of your ligand, and it will finally tell you on where exactly this your compound can bind to what protein. Okay, so this is what we have done. Now let us begin with our next topic. Like once we have identified the target protein, okay, by whatever method, by experimental method or subtractive genomic method or pharmacophore mapping method, you should end up with identifying a target protein for your disease. So say for example you have identified a target protein. The next job for you would be to check out if the three-dimensional structure for that protein is present or not. Okay, so the most important resource to find out if the structural information of your protein is present or not, by that I mean if you want to figure out if someone already has crystallized the structure of your target protein, you would go and look into an online resource and online database called as Protein Data Bank, what we call it as PDB. So this PDB is a database that is responsible for hosting the structures of experimentally solved proteins. Okay, so people usually go and search in this PDB database to figure out if the structure of the target protein is present or not. So now if the structure of target protein is found to be available in PDB, let's say for example if someone has already solved the structure experimentally and deposited it, then you can proceed for the next proceed for the next step, that is you need to then identify where exactly might be the binding site of your drug, or if it is a catalytic protein, then you can proceed for finding out where might be the active site of your protein. This step is included in active site identification. Okay, so now if you figured out ki, okay, the structure of my target is not yet been experimentally solved by X-ray crystallography, then there is no worry. You have kind of alternative way of solving the structure. Then what you do is that if the structure is not solved and somehow you are compelled to work on the same protein, you can use another computational tool that will yield you the structure of your target protein. So that method is called as homology modeling. So in this method, you look for the nearest homologue and then you deduce the structure of your protein whose structure is not solved utilizing the computational approach. So, well, let us first discuss what are the steps involved in homology modeling. That is the method in which you can develop the structure of your target protein if its structure is not experimentally solved. Okay, so here are the steps that are involved in homology modeling. So first step that you should be doing very carefully is that you need to identify a suitable template for your uh, protein target, or what we call it as target protein. So now the template is usually the homologue of your target protein. Okay, so now let me once more explain in more details on what exactly a homologue is. Well, say for example, you have selected or you have ended up identifying, well, say for example, any protein or enzyme from your leishmania as as drug target. Well, say for example, you have identified that any enzyme like homoserine kinase as an drug target from your leishmania, and somehow if you find out in go and search out in PDB and you realize that the structure of homoserine kinase from leishmania is not available, no one has experimentally solved it, or the crystal structure of this homoserine kinase from leishmania is not available, then what you would do is that you would try to find out a suitable template for your sequence. Okay, so target sequence would be the sequence from homoserine kinase of your uh, of your uh, of your organism leishmania. The target sequence would usually come from the pathogen. Okay, in our case, for sake of our simplicity, we have considered it as leishmania, and we have selected the target homoserine kinase. So usually the sequence of homoserine kinase is available. Okay, so it is necessary to involve, or you can say in order to build up the structure using homology modeling approach, it is necessary that you at least have the sequence of your target protein. Okay, so now let us assume that we have the sequence of homoserine kinase from your leishmania. Okay, so that would be the target protein whose structure is unknown. Okay, then next thing that you need to do is to find out a suitable template. Okay, so now template should be the structure which should be the homologue of your target protein. So now what is homologue? Homologues are the proteins which are evolutionarily related to the given protein. Well, say for example, our objective now is to find out, or is to deduce the structure of the target, that is homoserine kinase, from leishmania. But the problem is that first you need to find out a suitable template. And what I said earlier is that the suitable template should be the homologue of your target protein. That is, it should be a suitable homologue. It should be the homologue. In our example, it should be the homologue of homoserine kinase. Okay, so now what you will do is that you would try to figure out what is the nearest closest known relative to leishmania. It can be trypanosoma. Well, say for example, someone has already solved the structure of homoserine kinase from trypanosoma. So now you have got the structure of homoserine kinase from trypanosoma, and you don't have the structure of the homoserine kinase from leishmania. So now, using the computational techniques like homology modeling, you use the structural properties, or you can say the structural information of the homolog, and deduce the structure of your target protein whose experimental structure is not available. So this process is called as homology modeling. So what you do first is that you need to follow this protocol. First, you need to identify the template structure. In our case, our template structure was homoserine kinase from trypanosoma. So now what you would do, since the structure of this protein is available, therefore its sequence will also be available. Therefore, what you would do, that you would try to align the sequence of your target protein whose structure is unknown with the sequence of the template. So the objective of performing such alignment between template sequence and target sequence is to identify which are the regions in your sequence that is highly conserved or that is present in your template. Okay, so once you have identified the region, all you need to do is to copy the coordinates of all the align amino acid residue to the amino acid residue in your target sequence. So first you would copy all the coordinates for your backbone, and hence you would first try to build up the backbone of your target sequence. Once you built up the backbone, then there might be certain region which are not aligned in your sequence alignment. Those region we term them as loop region. So you separately built up the backbone of the loop, that is what we call it as loop building. And eventually, after building up the backbone of loop region, now you have the entire backbone of your protein molecule already built up. So next what we do is that you attach the side chain, you perform side chain optimization. After performing side chain optimization, you perform energy minimization. Okay, and your model would be ready. So once your model is ready, then you go on evaluating the model. One of the best way to evaluate the correctness of your model would be in terms of evaluating its stereochemical property. So programs like ProCheck implement the concept of Ramachandran plot to figure out if the amino acid residues are properly placed in a well balanced stereochemical environment. Okay, so you would create Ramachandran plot for your model, and eventually then in your Ramachandran plot you would check if more than 90 or 95% of amino acid residue should be present in core region, or maybe allowed region, or generously allowed region, and very few of the residues are allowed to be present in your disallowed region. Okay, so the thing is, when you are sure okay, my model is correct, then you proceed for active site identification. Now if you think that model is not right, then maybe you can continue with one or more steps that I have already mentioned before, and yield with the homology model of your target protein. Okay, so the software that are used to generate such homology models, one of the most important and freely available software is Modeler. Modeler is one of the gold standard used for homology modeling. Okay, so it is a standalone software. You need to download it. It is based on Python. So you would have to download Python first, and then you would have to write some Python script and run this modeler through those Python script. Well, it is very easy. Another option is that you can use online tool for homology modeling. So this Swiss model is one of the most popular uh, software, online software for generating your homology model. It would ask you for the sequence of the protein whose structure is to develop, and it would ask you for the structure of template, and it would perform all the steps automatically and give you the result in form of 3D coordinates of your 3D coordinate of your model of your target protein. Okay, so by however method, either you have the crystal structure or experimentally solved structure of your target protein, or you have yourself constructed the structure using the computational methods like homology modeling, you would end up with the structure of your target protein. Okay, now what next? The next important aspect would be to identify where exactly would be the active site of your protein. Okay, so where exactly your ligand is going to go and attach into that protein. So there are theoretically three main approaches for identifying the active site. So these three methods, I would just mention those, are evolutionary trace approach, reference ligand approach, and surface analysis approach. So I will explain you one by one in more details on what is evolutionary trace approach, what is reference ligand approach, and what is surface analysis approach. So the objective of using all of these approaches is to identify where exactly might be the active site in my protein model, in the protein model of my target protein that I have identified. Okay, so now let us talk about the evolutionary trace approach. So in this evolutionary trace approach, well, it relies on the fact that almost all protein functional sites, which include your active site, arise through mutation and selection, and therefore they will be one of the most highly conserved region in protein. Okay, so what usually happens in this approach is that you try or you begin with alignment of a lot of sequences, and what you do is that you try to figure out in those multiple sequences which are the most conserved region. So by the conserved region means is that you can start out with well, let us continue our example of homoserine kinase. So what you would do is that you would try to collect the sequence of homoserine kinase from different closely related organism. Well, say for example, this is homoserine kinase from Leishmania. This can be homoserine kinase from Methanococcus genesis. This can be homoserine kinase from Trypanosoma. This can be homoserine kinase from Plasmodium. It can be homoserine kinase from different species. So ultimate objective is that you collect the sequence of the target protein from multiple species and then perform multiple sequence alignment in this way, and then try to figure out what are the sequences that are conserved throughout all of the homoserine kinase sequences. Okay, so as you can see over here, well, over here whatever is highlighted in your red font, or whatever residues that are highlighted in red color, well, those are the residue in your active site. And you can see over here the residues in active site are present in the same order and are the same residue in all of the homoserine kinase in different organism. So in this way we say that this sequence of active site is conserved across all of the species of the protein called as homoserine kinase. So this is exactly what we mean as conservation. Conservation यानी क्या? Certain sequence, certain amino acid residues that are present in same order, same kind of amino acid residues are present in all of the sequence of the same protein in different species. If such is the case, we say that the sequence of this active site is highly conserved. So, well, now it is very logical to keep the same residue in all of the protein. ये देखो इसका conservation रहना जरूरी है. Why? Because being it a homoserine kinase, that means it has to perform certain function, right? And in order to perform certain function, it needs to create a specific chemistry in its active site. Okay, and this specific chemistry can be only created by having certain specific side chains that are supposed to be present over there so that it can perform certain function. So the same chemical reaction is needed to be catalyzed by the enzyme, whether it is present in one organism or another organism. Therefore, usually the same type of amino acid residues are present in the active site of most of the protein, of most of the related protein as such. Okay, so what we do, well, there are programs like Crescendo that exploit this approach. Okay, so what such programs do is that they ask you for the sequences of your, well, so for example, your target protein and the related sequence from different species, and they perform multiple sequence alignment in such a way, and then they highlight ki, okay, these are the regions which has got highly conserved sequence. Okay, and finally what you do is that next you map those sequences onto the structure and check out if they are present in its active site. Okay, so this is how you identify the active site of your protein through something called as evolutionary trace approach. So over here you can see this red color font or red color amino acid residue that are shown over here are present right in the active site of this protein. Okay, so besides active site, there might be other functionally important regions in your protein. Well, say for example, there are upper and lower lips in this homoserine kinase. You can see over here; those are colored in cyan residue. Those are colored in cyan color. So the function of these two structural units is to close and open the enzyme when the substrate attaches or when the catalysis is over, when the product needs to leave that enzyme. ओके तो ये अपर और लोअर लिप का भी सीक्वेंस काफी कंजर्व्ड होता है. बिसाइड दैट देयर आर डिफरेंट मोइटीज. वेल से फॉर एग्जांपल सिंस इट इज अ काइनेज, you have got motif 3 that is responsible for accommodating your prosthetic group ATP or ADP. There might be certain another motif that is responsible for certain another function. In this way, along with sequence, there are certain structural motifs that are also usually conserved in your protein. Okay, so in this way you identify the active site based on the conservation of your uh, amino acid residues in its sequence. So this is how you try to figure out what is the active site of protein using the evolutionary trace approach. Okay, so the structure or the example that we have discussed, we have recently published it in this Journal of Molecular Modeling. You can read this paper for more details as such. Okay, so the next approach is called as reference ligand approach. So now in this reference ligand approach relies on the fact that we already know that most of the ligand, well, this ligand can be either inhibitor if it is a competitive inhibitor, or substrate usually goes and bind to the active site. right? I hope you are comfortable with this concept. Ki most of the substrate, or if it is a competitive inhibitor, it would go and attach to the active site. Therefore, in PDB, most of the protein structures are those who are co-crystallized with such ligand. Next, what we do is that we identify such structure, we identify our ligand, and we identify what are the residues that are present in close vicinity of our ligand, whether it might be substrate or whether it might be your competitive inhibitor. So the amino acid residues that are present in its close vicinity, those are interacting with ligand through their side chain, are listed out as active site residue. So once you figure out, well, say in this case of homoserine kinase, this is your substrate molecule. HSE stand for homoserine kinase, and in its close vicinity, you can see there are proteins like aspargine 145, aspartate 144. You have got arginine 241. Then you have got one aspartate, another aspargine. So all of these neighboring amino acid residue interact with your substrate through their side chain. Therefore, there is a strong possibility that these side chains might play some very important catalytic role. And based on this assumption, we would declare these proteins as these amino acid residues as active site residue. So once you have located what are the amino acid residues that are present in vicinity of your ligand and they interact through their side chain, you might then further dig into literature and confirm whether whatever amino acid residues that you have identified are active site residue by maybe checking what is the mechanism of action of that protein, or what is the catalytic mechanism of that protein, and if these residue fits into that role. And in this way, this is the best way for identifying the active site in your protein. Okay, so there are programs like FlexX and Moligro that utilize this reference ligand approach for identifying the active site. Well, what you have seen here is the active site of the enzyme called as homoserine kinase from Leishmania major. You can read out this paper for more details of how this protein function and other details that you can find interesting. Okay, so third and final approach is something called as the surface analysis approach. Well, according to me, it is not as reliable as compared to the previous two approach. Uh, the previous two approach, like your reference ligand approach and your evolutionary trace approach. Well, there are reasons that I would explain you now. Okay, so first let us try to understand, using the surface analysis approach, how do we end up identifying active site first. Okay, so this approach make use of the fact that most of the active sites are the deepest cleft in the protein surface. Okay, so this is the assumption, the basic assumption based on which surface analysis softwares identify the active site. So what exactly happens here is that you obtain a protein. The next thing that you do is that you create its surface representation. After you create its surface representation, you identify where exactly and how many clefts and cavities are present on your protein. Okay, on that surface, how many clefts and cavities are present. Then what you do is that you measure the surface area and volume of each and every cavity that you have identified. Okay, then you map all those cavity again on your protein surface, and depending on what cavity has got the deepest cleft and which cavity has got the maximum surface area, you would declare that that pocket as an active site. So softwares like Pocket Finder and QSite Finder are two most popularly used online servers that take PDB file as an input, and they detect the active site by means of surface analysis approach. So now let us discuss the fact on why I am hesitant on using this surface analysis approach for identifying it as an active site procedure. Okay, so before I justify myself, you should understand कि what exactly is an active site. तो आपको ये समझना जरूरी है पहले कि एक्टिव साइट एग्जैक्टली क्या होता है. So active site are usually that small portion in your protein wherein specific functional group orient the side chains in a very precise manner that would create a functional chemistry for that protein that would enable that protein to perform certain catalytic function. Okay, so that is how you biochemically define an active site of that protein. Okay, so in active site, there is always some chemistry that is involved, right? It is necessary that certain functional chemistry is created in that small region of your protein that is responsible for catalysis or even inhibition. So now whatever surface analysis approach that I have just mentioned, it do not involve any kind of chemical moieties, or it do not involve any kind of chemical environment that is considered for declaring it as an active site. Okay, all it does is that it tries to identify the deepest cleft with maximum surface area and declare it as an active site. So, well, it is not necessary that the deepest cleft is always your active site. Okay, so well, say for example, what if you have started out with a monomer of a protein which you know forms the active site when it get dimerized, and this active site is formed at the interface of the dimer, right? So in that case you would never end up with identification of proper residue, right? So whatever deepest cleft it has identified on the given input structure, it would calculate all of the cavities, and whatever cavities that it finds out to be the deepest cavity and having the maximum surface area, it would report it as active site. So my point is that being a biologist or being a biochemist or being a chemist, it is your responsibility to make sure that whatever output that your computer program gives, it is your responsibility to make sure that it is authentic, to make sure that you compare it with say similar studies and make sure that you are going in a right direction. Okay, so now this is how things are done on QSite Finder and Pocket Finder. This figure, you can see that it has identified numerous site like 10 site, and for each and every cleft, or and for each and every pocket, it would calculate the maximum surface area, and it would calculate the volume for each and every cleft, and the cleft, or you can say a cavity that is having maximum surface area and maximum volume, it would indicate it as your active site. So it is all up to you now to decide which type of approach is to be used for identification as an active site. Okay, so this is it. Okay, so now you have identified a target protein, you have somehow identified the active site, but you cannot directly proceed for molecular docking or virtual screening of your compound. There are certain few steps that are still needed to be performed in order to make sure that you have prepared your protein properly. Okay, so I would just briefly explain few of those steps that are essentially required for preparing your protein for further virtual screening process, and all of these procedures are included in something called as target processing. Okay, so in this section, in when you start processing your target, you make sure that uh, if your protein is well, say for example multimeric protein यानी क्या, it might have different chains, multiple chains, and it is entirely possible that in a single chain, if your active site is present, then there is no need of keeping another chain in your structure, right? So what you would be interested in is to sort out the subunit if it is a multimeric protein that contains your active site, right? So if your target protein contains more than one chain, you are advised to focus, or you are advised to isolate or separate that chain which contains your active site. Okay, so once you have isolated or once you have separated the chain that contains your active site, next what you need to do is that there might be some unnecessary components, like there might be some solvent molecules which are present around your protein. Then there can be crystallographic artifacts that are usually used for improvising the crystallographic condition, and somehow they also get co-crystallized and they also appear in your protein structure. So such unnecessary components are you can remove them. Okay, but unnecessary component are supposed to be decided priority wise based on what type of system you are dealing with. Okay, so say for example, along with your substrate or inhibitor, if you have got prosthetic groups or cofactors which are essentially required for the catalysis of that protein or inhibition of that protein, you are supposed to keep it then. Or if you figure out कि okay, I have active site in my protein at a position which is far away from where the prosthetic group goes and attaches to itself, or it differs significantly from the site where cofactors are attached, then you can remove the prosthetic group or cofactors. Okay, so you need to decide which of your component in your protein structures are necessary and which are the component in your proteins that are necessary. So that would vary from case to case. You would have to dig out into literature, you would have to read out certain papers and make sure that you are not removing things that are very important for the activity of that protein, right? So many times it happens that your inhibitor stays in close association with prosthetic group like PLP. It might require PLP, or it might require ATP, ADP, or even heme group. So all these prosthetic group are essential for the activity of that protein, and you cannot remove them if they are essential, right? So my advice for you is that you first decide कि whatever prosthetic group are there, where they are present, are they really important for the activity of my protein? If they are important, retain them. If you feel that those are not important, you can remove them. So this is very important. What people or what students I have seen usually do is that they remove everything from that protein and end up with say some apoform of protein that they use for molecular docking. Okay, so just make sure that you retain the important things and you get rid of things that are not important. Well, that is what things are to be done in the step of target processing. Okay, so once you have removed the unnecessary component, next you check if your target protein has got chemical ambiguities. So say for example, if you are using an experimental structure that has been solved by X-ray crystallography, then if in your protein, if there are certain regions which are involved in high mobility, well, say for example, there might be certain loops which are always involved in motion in your protein. So such high mobility loops are usually not resolved in your electron density map, and hence they do not appear in your model. Therefore, you need to first make sure that are there any important region that are unresolved or that are absent in your proteins. So if they are absent, you need to build up those. Okay, or maybe select certain another model which has got those crystallographic unresolved region. Okay, you would have to also make sure that it is entirely possible that there might be alternate, or you can say multiple coordinates for a single atom. Okay, if that is the case, then you might want to retain only one of them. Okay, you would also have to take care of hydrogen torsions if there are. Okay, and finally what you need to do is to you need to add up the hydrogen atoms. As you know that if you are using protein models that are experimentally solved by X-ray crystallography, the hydrogen molecules are not usually resolved, and hence they are not detected in model created using X-ray crystallography. Therefore, in target processing step, the softwares usually add up the hydrogens. Okay, then what next thing that you should take care of is that you need to optionally check if the charges in your active site residues are optimally normalized or not. Okay, so checking the protonation and torsions of amino acid in active site carefully is very important. Why? Because chemically it would make a large difference whether an amino acid residue act as a donor or as an acceptor. Well, say for example, I would like to cite an example like if your active site contains say acidic amino acid residues like aspartate, which would contain your acid group COOH, or it might contain arginine, which might contain your guanidino group, right? Your guanidino group contains a lot of amines. Okay, your aspartic acid contains acidic group. And what happens at physiological condition is when the catalysis occur, the active site residues might lose the protons and form uh, aspartate chemical moiety. Okay, aspartate chemical moiety is the moiety in which your aspartic acid has lost the proton, and we call it as aspartic acid when there is OH present in your side chain, right? And aspartate is that when O negative is present in your active site. Therefore, the presence or absence of that single hydrogen would enable that aspartic acid residue to act as either donor or to act as either acceptor, right? So if there is an hydrogen present over there, it would act as donor. If hydrogen is not present and it has got negative charge, then it can act as acceptor. Therefore, it is all up to you to detect in your active site what residues are involved and what chemical environment is necessary for the for proper functioning of your protein. So you need to optimally place charges into the active site of your protein. That is very important. Okay, the next important thing that you should take care of is you should take care of the tautomerism that usually happens at the active site. Okay, so it is I'm sorry, it is what is tautomerism? Tautomerism, you can define it as it is a natural phenomenon in which there is shift of hydrogen along with the path of alternating single or double bond. Okay, and such shifting of single or double bond is mostly involved in, around nitrogen and oxygen. Well, so for example, in case you have got say histidine group in your active site, and you know histidine group has got an imidazole group present over there. Okay, उसके साइड चेन में क्या होता है, imidazole group होता है. और ये imidazole group में it involves shifting of double bonds. Okay, so where exactly that double bond is present? So this histidine is it has got alternative tautomeric state, and we usually denote it as HSE HIS or HSD sometimes. So you would have to make sure that where exactly that hydrogen bond where that double bond should be present. So you would have to take care of tautomerism as well. So now next question, how these things are done. So whatever software that you use for molecular docking or virtual screening, they have got its own inbuilt utilities for target processing. All you have to do is to make sure that things are all right. Everything, every utility is usually present in your software. Okay, but you should be aware of it. As an informed user, you should know what is happening in each and every step. Are things going in the right direction or not? Okay, that as a biochemist or a molecular modeler, you should always take care of these things. Okay, so now say for example, for commercial packages like Gold, Moligro and Fred, so they have got their highly sophisticated software or the highly sophisticated section that take care of this. So there are some free softwares like Argus Lab, and there are some GUIs which are developed by MGL laboratory, molecular graphics laboratory. Well, these GUIs like MGL tools, like Python prescription, they are used for molecular docking using their own suite called as Autodock. Okay, so they can be utilized for the purpose of your structure preparation. Okay, so besides this, you can also use UCSF Chimera to process your target if you are planning to use dock software for molecular docking. तो अगर आप ऑटोडॉक यूज कर रहे हैं, if you are using Autodock, then I would advise you that you use one of the GUIs like MGL tool, or you can use Python prescription, or you can even use Vega ZZ for running that Autodock in the background. Okay, and if you and Autodock is an open source software, you don't have to pay anything for that. So is dock software, and you can use UCSF Chimera for target processing, which is also a free software. So by the end of this step, you would finally get a complete protein structure whose active site you have already identified, and you have already protonated it properly, you have already placed a proper charges, you have made sure that it contains all of the important components that are necessary for either activity or inhibition of that protein. There are no crystallographically unresolved region present in your protein, and everything is fine. And finally, you say that you have got your target protein right. Okay, so this is how you end up creating a target protein. I have just mentioned what are the important things that you should need to do before you proceed for further molecular docking or virtual screening. Okay, so next in our next lecture, we would be talking about how ligand library is obtained. What are the things that you should do before using it for virtual screening? Like how do we filter that ligand library and all the related stuff. So see you next time.